We've got a panel discussion coming your way on FAIR data for successful digital transformation. And I'll quickly introduce our panelists. Hugh Salmon is Director of Bioinformatics Scientific Strategy at Exelixis. Gautam Sridharan is a Principal Scientist with Alnylam Pharmaceuticals. And then Kishore Nandiala is a peer of Hugh's as the Executive Director of Digital Transformation, also at Exelixis. So all four of these folks have led transformative FAIRification efforts within their enterprises. Panelists in this talk will talk what the infrastructure to handle biomedical big data should look like, the challenges they faced in the real world doing this while adopting FAIR approaches to data management. And then they're going to talk about some wins that they've experienced in this journey to FAIR transformation. And then we're going to be emceed by John Conway, who's with 2015 Visioneers, and he's going to moderate the discussion. So John, take it away.
Great. Thank you very much, Bobby. Can you guys hear me okay?
Yes.
Okay, great. All right. I think it's important to point out here as well is that we have different personas in the as panelists. And I think it would just be really good brief introduction, maybe just take 10, 20 seconds for each of them to kind of say what they do, you know, where they work and kind of their view on FAIR data and digital transformation. So why don't we start with Kishore, and he can go first, and then we'll go to Gautam and then Hugh.
Yeah, thank you, John. Yeah, my name is Kishore Nandiala, part of Exelixis digital transformation function, part of the technology group here at Exelixis, and my remit is supporting both drug discovery and drug development. So that's a quick intro about myself.
Great. Gautam.
Thanks, John. Gautam here, scientist at Alnylam Pharmaceuticals. I sort of lead a multi-omics group. Predominantly, we lead proteomics and metabolomics efforts internally, where we generate data using LC mass spec, and we do a lot of bioinformatics downstream processing as well. And we face a lot of challenges in integrating those multi-omics data sets and delivering quality reports for our biologist colleagues to interpret that data. And so we think about FAIR data principles all the time in terms of integration and modeling. Sorry, I'm on my phone. My laptop suddenly crapped out for no reason.
No worries. It looks fine. Great. Thanks, Gautam. And Hugh.
Thanks, John. So my role at Exelixis in scientific strategy is to really make sure that lots of omics data and data science, data analysis actually comes to bear on decision support for evaluating mostly biotherapeutic targets and strategies, mechanisms. And so the challenge is to bring a lot of data very quickly to data analysis and then simplify in the support and discussions in teams. And so that's actually a massive challenge. There's a lot of really interesting data out there, but how do we get it integrated quickly? And I've had experience in the past doing this, so understand how important it is to have data that is fair.
Yep. Okay, great. First question here, and basically, you're all kind of have different personas, different responsibilities, but the key question is, how do you assess FAIR data? How do you know that you're on track? How do you know that you're not behind or you're right on target? And so the audience knows, we spoke about this briefly beforehand for a couple minutes, and everybody might have a little different view on it. But the most important thing is to say, from the different persona perspectives, what their thoughts are, but also how do they articulate that, how they fight for more, how do they make a case to the company or organization they're in to say, hey, we need to do more, we're right on track, we're doing great. So I'll start, Hugh, since you were the last one to talk, I'll let you go with that first.
Okay, so I think from the perspective of data analysis, you know that something has gone wrong with your data when it's difficult to integrate it, when it's costing a lot of effort, it's causing delay, when you have to seek harmonization on the fly, as opposed to having things already harmonized, then you know there's something wrong with the environment of your data. It's not fair and it's not harmonized. That's really where I think one can measure the problem. As far as showing the value, I'd like to take an analogy. If you invest in sequencing or a fax center, you don't think you're going to be saving money. You know you're spending money, right? Why do you do that? You're going to be supporting some decisions with whatever's measured there. I think the same thing applies to data integration and storing data better and being able to make it accessible. You invest in it. It's going to cost money. We shouldn't pretend it's going to save time. It's going to take time. It's going to cost money. You do it because you're going to have better decisions down the line. And that's really important in pharma.
Okay, great. Thanks, Hugh. I'll just go back in reverse order on my screen. So Gotham, if you, as a scientist, you have really strong views on this as well. It may be a little different, but great to hear. Yeah,
for sure. For me, when I think of fair data in the omics space, the most important thing for me as a scientist in computational biology and systems biology is the data findable and is it well annotated? And, for example, we may have hundreds of RNA-seq, proteomics, and metabolomics data internally on platforms that look at publicly available data sets. But simple queries need to be seamless. Like, for example, let's say I want to know, I have a particular target in mind, gene target, and I want to know every data set or perturbation in which that gene is up or down regulated. That needs to be a seamless process. The controls need to be well annotated, perturbations need to be well annotated. And, I think as an organization, as we grow, we have to think about what do we need to keep in place for that kind of culture to grow, where everyone does annotate their data in a way where it is findable and we adhere to those fair practices. So I think that's what's most important to me in thinking about that biological context.
That contextualization of the data that you're talking about is so critical. We try to show people through examples and stuff, but this isn't like, your parents or grandparents' drug discovery anymore. This is like high volumes of data, meticulous data, the whole multi-omic space, and it has to come back together and fit together like a puzzle. And if you spend all that money generating that data and it's not contextualized, it's not fair, you just kind of hit a wall, right? You're going to hit a wall with your data and you're not going to get the results that you want to. So that's like a big, for me, and I think Gotham, you just said it's a big take-home message that you have to put almost as much effort, or a 50-50% effort, in your data strategy and your data environment, along with your experimentation, for multi-omics to really pay off in insights and productivity.
Yeah, completely agree. And it's not just the omics data. We look at, obviously, disease models and models of disease progression, models of disease amelioration, multi-omics analyses on these models to come up with, signatures that we can use for prediction as well as biomarker discovery. So that's basically the large scope of what we do. But oftentimes our colleagues will want additional information. So, for example, based on these few markers of disease progression, which ones are expressed in a particular tissue? Which genes are enriched in a particular tissue? And then you may want to bring in additional data sets. For example, I think many of you in the audience are probably familiar with the GTEx resource from the Broad Institute, which gives you tissue specificity of genes. So that needs to be integrated. And, someone may ask, well, can we figure out which pathways these all belong to? And so all of a sudden, you've got so many different layers that need to be well integrated. And that's not possible unless you have some sort of governance in place for what data needs to look like, how it needs to be annotated, how it needs to be organized, and what the end user actually needs for a particular application.
All right, thanks. Sounds great. And Kishore, you want to talk from your vantage point from a digital transformation perspective?
Yeah, so again, I think our focus has been more so, how do we best support, multiple functions, right, within the same company that have bioinformatics groups. So our focus is a bit more sort of at a higher level. So for instance, if Hugh is doing something within scientific strategy and we have a different group within drug discovery, how do we make sure if he's ingesting data and curating data that is easily accessible by other, or at least first known to other functions. Sometimes different organizations are running, internal organizations are running at such a fast pace that it's not always visible, not by design, but it happens. So our focus has been more around how do we make sure that we have sort of the enterprise view of things working with different functions. How do we make sure that we're bringing people along using technology or streamlining and standardizing the platforms that we use. For instance, Poly as an example is one of the platforms that we're embarking on, where Hugh has been using it for many months, and hey, how can we expand that so that there is synergies in terms of what is being done. So
okay, so stay on the line because I'll ask the next question, and if you want to or can answer it, you'll go first. And it's really about the examples of stakeholder management, alignment, scientific data strategies. Do you have like lessons learned when it comes to that and getting this this fairness and digital transformation off the ground, and how it went about? Any lessons learned to share?
Yeah, so I can certainly share what we've been doing thus far. Granted, we're in our early stage in terms of our journey thus far. I'll maybe start off with some of the challenges that we've had, right, when we started to go down this path. I mean, lack of alignment, not clear clarity in terms of why people are doing what they were doing, right? And it's not anything, not to take away anything from anybody, but that's how it all started. And then after 2 or 3 months, we disbanded. There was a group that was meeting pretty frequently, and people felt like, hey, I'm not getting anything out of this. So we took a step back, again, met up with each of the stakeholders individually to understand what their concerns are, and sort of made it a bit more meaningful. So now we have a structure where we meet once in 3 weeks. It's a cross-functional team, and it's really sharing of information, what type of data sets, what type of tools that these teams are leveraging. And then if there are follow-ups that need to be had, at least it's a safe environment for people to share what they're doing. So that, again, Hugh can chime in, and he's one of the reps who's part of that cross-functional team. I think it seems to be working well. In the end, I think people have the right intent, but every functional group has their own pace at which they want to deliver, what they're supposed to do. But if we create an environment that's safe and create, platforms that are reusable and interoperable, I think it's something that seems to work for us.
Yeah. So Hugh, why don't you go next? But it sounds a little bit of change management and what's in it for me, or what's in it for the end user, and how are they, how are they going to get started? But go ahead, Hugh. You're on mute. Hugh, you're on mute.
Indeed, I am. Sorry about that. What we've really been coordinating is technologies and taking those first steps into trying to understand why we make decisions about, is a technology actually going to be helpful? Is it going to further our ambitions to be fair or not? And those are important decisions. And so, the Poly platform is a step in a great direction for us. When it comes to actually thinking about fair longer term for an organization, I think that governance is absolutely key. And you've got to have, it's not just stakeholders, you have to have intense discussions between people from completely different disciplines. You've got to have bioinformaticians in there. You've got to have biologists sometimes. You have to have clinical folks, and you've got to have engineers who really understand, how you're going to implement dictionaries, ontologies, taxonomies, provide templates so that bioinformaticians actually using what they should be using when they're ingesting data so that it complies with FAIR. So it's actually quite an undertaking, and we really are just starting. But I think it's, it requires a level of organization that organizations don't want to invest in yet.
That's a really good point.
Yeah, and I've done this from outside organizations as well, where you're less invested individually, and it's still difficult and requires a lot of patience to get people to put that extra work in. But I actually think it's fun work. So I try to find people who also agree that, there's something beautiful about making data all work together.
Yeah, the return on investment, honestly, for everyone as a whole is a paradigm shift for the world, right? But could also be a game changer for the organization. So it's just like any other policy or public policy change. We all know those take 5 to 7 years and stuff. And the companies that started, some may get done earlier than that, but the large organizations might take a little longer to get through this. But the return on investment will be immense. That's my prediction. And then Gotham, you might have a different view on this, right? You're the scientist. You're doing a lot of the experimentation and the analysis and stuff. So what have you seen from your perspective?
Yeah, what I've seen is that there's a big benefit to having a domain expert early on in the data governance process, giving their feedback. I think it's tempting for a company to be like, okay, this is an IT issue. It's not an IT issue. It's it involves everyone. And as we think about questions like what does everyone have to input when they upload their data set, that needs to come from a biologist who understands the nuances of the experimental design, what the dosing strategies were, what the different conditions that are relevant to a particular study are like, and that will go a long way in doing the type of data integration that we're discussing in this panel. For example, a lot of times our colleagues want to know if in a particular system, if transcriptomics correlates with proteomics, does gene expression correlate with protein level? That's not a trivial ask. There's a lot of nuances in how the RNA-seq data are collected. There are a lot of nuances in how proteomics data are collected. The two are very different. One is counts based on a sequencer. One is based on raw spectra from mass spectrometry, and the way fold changes are calculated are very different. The way you in proteomics data, you get a lot of lowly detected, basically not a number of values, and filtering your data set for zeros and all that. It's also very challenging. So understanding all those nuances at the early stage requires a domain expert. And then I think you need to have that conversation up front to make sure you're adhering to some FAIR principles and that goes a long way.
Yeah, no, absolutely. I mean, the variables, just the number of variables, degrees of freedom, you just go down the line, are exponentially increased, right? And so if you don't have that domain expert, and you're not trying to capture the essence or the most of whatever you can, you probably are going to run into this situation where you're like, oh, we should have captured that, that metadata or that data, because now we're at this point where we're uncertain if this data can even be aggregated or analyzed together, et cetera. So now it's awesome. Okay. I might combine the next questions, the cadence to becoming FAIR. So what steps, and it kind of goes back to the alignment part, but if maybe Hugh, I'll start with you. If you said, hey, these are the 3 or 4 or 5 steps we took to get this process going, what are those high level steps, if that even makes sense? If it doesn't make sense, I'll ask a different question. And you're on mute too, just say.
I'm muted again. I don't think we've taken enough of those steps to really give you a helpful answer for that yet.
Right.
I think that I just quickly borrow on what we've been hearing. You've got to get the right people in the right place, and then you have to give them a mandate to actually decide how things are going to be. It's essentially about aggregation of data, right? It's storage
Right.
with the purpose of aggregating. So you've got to give them some authority, and they've got to in return provide something. Policies without tools generally are ignored.
Right. Okay. Let me ask this question then. Data standards. Where do data standards come into your strategy? Is it part of your strategy? If it is, how are you able to drive the adoption of data standards?
Again, in this situation, haven't done it. In previous situations, it's really about making sure those templates and tools are in place, and that you have the flexibility. You thinking from a software development point of view as well. It's like, well, if we had to do this a little bit differently, could we do it again? Can we afford to do it again? Because as you go along, as you say, you'll get somewhere and it's like, oh my goodness, nobody thought that was important metadata. We forgot it. You want to be able to bring that in quickly and execute. So it's as much about making sure your tools are FAIR compliant as your data are. And that's actually a very interesting area where you actually get software developers to appreciate data. And so I think it's often good. Sometimes that's from the bioinformatics point of view. I think it's actually even better when you have real infrastructure coding people in place to help.
Okay. Hey, Gautam, this could be a great question for you too, if you think the data standards, putting adoption of data standards on a scientist. I'm sure you could see pros and cons to that. So could you share some of that? Like, hey, from a pros, yeah, this and that, but from a cons, it's a time, warp, it's this, it's that, but maybe your thoughts on data standards and adopting them.
Yeah, I think, if we want to do any sort of predictive modeling, at our company, we're an RNAi-based therapeutics company. So obviously we do a lot of modeling for how effective are drugs at silencing targets. And you can imagine there's a lot of different conditions for that, different doses, different cell lines, different, whether it's in vitro or in vivo, whether or not, the type of target that we're hitting. Sometimes those targets may have, different mRNA turnover, protein turnover. So there's so many different variables, and doing any sort of predictive modeling is ineffective unless you have some standards in place to put those variables in a meaningful way. So I think having those in place like we discussed this, go a long way. And of course, there's a little bit of a bottleneck there because, if you're a lab scientist, putting in 8 hours at the bench, the last thing that's on your mind is like, oh, I need to go fill out all these forms and online templates and whatever it may be to make sure my data are fair. There's still a little bit of a cultural barrier where a lot of people are still comfortable having their own templates and own things on spreadsheets that they've created for a particular niche experiment, and then upload that spreadsheet to a cloud platform. Because and I think that's from a data integration and data science perspective, that's the worst case scenario where you have a lot of data that's stuck in spreadsheets on the cloud that can't be integrated in. But I think a lot of that is just a little bit of a communication gap and people coming from different backgrounds, right? Maybe a hardcore biologist just doesn't appreciate the necessity of having data structured in a certain way, and having these kind of panel discussions and it basically conversations go a long way.
Right. I think Hugh just made a comment. The reward to scientists for standardization can be that data analysis correctly entered data can be better analyzed. Totally agree. Here's another thing that I'll share with everyone is whenever we did that iterative process before we implemented an ELN, a LIMS system, a bioinformatics system, whatever, the adoption was 75% faster and better. And it's eye-opening, but I have to tell you though, a lot of our clients push back on doing that work up front. They want whatever solution is, they want it implemented next week or next month or in the next 2 months. And we're like, no, we need to do this first. And they really argue with you and push back and sometimes threaten to fire you. And, you know, you get put in this position. But my experience over the past 30 years is when you put that in place first, you have blinding success in your implementation and the journey that you go on with that scientific software solution. So, okay. Let's see, what's another good question? And by the way, you guys can ask a question too. It doesn't just have to come from me. So if someone on the panel has a good question to ask everyone, please ask it. But whenever you want. Here's one question around where do you stand in the current investments for FAIR data management? So are you investing in data curation tools? Are you investing in data scientists? Are you investing in data management software, et cetera? So, okay, Gotham, like, again, this may not be the best question for you, and if it's not, we can go to someone else. But from an investment perspective, and you know the tools that you have to use to curate data and to data, you know, data management, etc. Is that something that you can speak to?
Yeah, I mean, I can take a stab at that. You know, we for us looking at all the different omics data sets in the public domain in one platform was very important to us, because that is, you know, what's in the public domain is very scattered. You know, there are no fair governance processes for geo data sets other than having it on the portal, which isn't very well managed. So, I mean, putting a plug here for, you know, Lucidata and Poly, that's kind of where we wanted to start. And it's like having a platform where we have access to these thousands of data sets that are organized and implemented, and using FAIR practices allows us to access data and interpret data in a way that's consistent across all the different, you know, studies that we're looking at. So I think having, you know, a platform is a start, right? And investing in that platform and believing in it. And then the next step is going to be is user adoption. I think that's a whole different challenge that you know, we can speak to maybe in this panel as well. How do you get people to use something? I think a lot of our colleagues are inundated with different tools, ELN tools, internal wikis, you know, different, you know, there's box, there's all sorts of different softwares that they have to know and understand and use. You know, is one more platform, what's the barrier to get someone to use one more thing? And I think that's going to be a challenge that we and others may face.
Okay, great. Hugh?
Yeah, and I think, I mean, maybe if
go ahead, Kishore.
Add on, John, is I think one of the things that we're trying to do, not just within the discovery area, but more broadly, right? You know, how do we sort of shield the complexity of technology, right? And again, if you were to look at the four walls of XLXs, there are a lot of things that, you know, happen, you know, whether it be your email or, you know, like Box or and so on and so forth. But, you know, oftentimes, you know, as technologists, we don't think the complexity that is being introduced as a result of a capability that gets introduced, and we don't necessarily do a good job of doing proper change management, right? And that's what leads to, you know, better adoption. So we are, you know, heavily focused on ensuring that anything that's being, you know, rolled out, there's proper communication, there's proper change management training, and understanding the personas within an organization in terms of what sort of tools and capabilities they use, and how do we make sure that as we're looking at newer platforms, that interoperability, you know, exists.
Okay, great. Hugh, do you want to add anything to that?
Sure, just a kind of last point. I don't look at the data ingested as the only data. All of the data analysis results are data, and you have to keep those there. And that's usually where people notice that things are not working, is after a couple layers of analysis, you can't integrate anymore. To that end, I've been collaborating with IT here to containerize and create an elastic compute environment that will provide the metadata that we need so that those results can be stored in such a way that they are fair. And that's really where I think a lot of the hard work needs to happen for bioinformatics, is not only do you need to ingest data, harmonize it, I guess try to cajole all those large organizations to cooperate over time, provide us data, but also to build internally tools that keep the philosophy going. And then when people see these really large, well-integrated analyses and meta-analyses that you can do on analysis results over time, I think that will spread the whole concept. So that's, if you think of what's really impressive sometimes in a publication, is they've really got a hold on a wide variety of data and they brought it together. When you can do that inside a company, I think people will notice, because it's a huge challenge.
Okay, awesome. So we're getting towards the end here, and I think what I would kind of ask is a pretty general question around efficiency gains that you've noticed from going to, through a digital transformation. It's a journey. I'm not saying anyone's done or even close to done, but real quick, Hugh, is your company seeing a jump in efficiency or a jump in participation, whatever it may be? What kind of change are you seeing? And we've got a minute, so if everyone can kind of give a 20-second answer.
So the 5-second answer is we're not there yet. This is early days. So we weren't improving something. Okay. And then just to take another couple of seconds, I did this in bioinformatic contracting company that I co-founded before, and that allowed us to provide much faster turnaround on projects by creating a system that harmonized data as it came in. And so when you're light, you can do it.
Right. And Gotham, the scientist quality of life, what are you seeing when you're able to do some of these things?
Yeah, I'm seeing that, things that would have taken a lot longer, for example, we're interested in looking at a particular disease model without saying what it is, and looking for all the different, transcriptomics data sets that are out there that pertain to that disease model, so we can look for different gene targets. And that would have been a challenging process before, because you would have to look at all these publicly available data sets, normalize them, figure out if they're correctly annotated, et cetera. But having a consistent platform that does that for you enabled us to take a project that may have taken months in just a few weeks. And I think that's a shout out to Lucid and the team. And I think we would like to expand that effort to different disease areas, different types of data, and integrate internal data with the external. So I think that's where we're headed.
Well, it sounds to me that it's been a significant improvement. So great job, everyone, including Elucidata, for writing great software and providing the team with what they need to get the job done better and faster. Thank you very much, everyone. This has been great.
Thank you so much, John, for leading this discussion. And Hugh, Kishore, Gautam, really, really stimulating ideas going around the table. Very much appreciated. So thank you again so much for taking some time. I know your busy schedules. Very much appreciated.
Thank you. And thank you, John, for creating.
Yep. Thanks, everybody. Take care.